special guest, who said he has made the Sasquatch. And here, I will now introduce a man whose name we can't disclose. Welcome, Dr. Undisclosed. So what I can see here on this is that you have made a portfolio that says you have made the Squatch.
Yes. Well, we went through, it wasn't so easy as we just made the Squatch, you know? We had to go through many different phases in order to perfect our final Squatch, you know?
I see.
The first phase we went through was selective breeding. We tried to do it the old-fashioned way.
Sounds cool.
And basically, for anyone who doesn't know, selective breeding is where we take an animal or an organism with certain traits that we want, and we pair it with another animal with the same traits in order to produce the desired effects.
Yes, I know that term.
Yes, indeed. So, in order to speed up the process, we try to use inbreeding, which is where we combine the two organisms from the same family in order to get the specific traits we would like. It works a lot faster than just regular selective breeding. But it also has side effects where we can bring out a recessive gene for a certain disease like colorblindness in humans.
Yes, bring out colorblindness. Sounds fun. Since I can't see color.
Oh yeah, I'm sorry. Yeah.
That's okay, please continue on.
Like I was saying. So then, selective breeding didn't exactly work since we couldn't find the monkeys or the primates with the correct traits that we would like together. So we tried manipulation. And in manipulation we used genetic engineering, which, for those of you who don't know, is where we basically edit the DNA of any organism. It's still, genetic engineering is still a work in process though, seeing as how we haven't perfected it quite yet. But in order to do genetic engineering, first we have to extract the DNA from the organism cells, and then we put it, we slice it up into smaller parts using restricted enzymes. And we put those small parts on a gel electrolysis station where we separate the parts from smallest to largest. And then we take the parts that we need and we put them into a sort of copying machine for DNA.
Sounds very scientific.
Oh, it is. That's right. And then we copied the DNA like 1,000 times in order to get the desired effects.
Well, are those all the types that you've done?
No, actually there's one more, Mr. Vader. It's called transformation, which is where we inject the DNA directly into the cells of the organism we would desire to have those effects. But that didn't quite work in the case of Sasquatch. We like to call him Bobo.
Why do you call him Bobo?
Because he's test subject number 8080. See, genetic engineering. Genetic engineering isn't quite perfected yet, as I mentioned earlier. So there's a lot of failed experiments and a lot of bleeding monkeys.
Sounds very bloody.
It's
Well, Mr. Undisclosed, I hear that you've had another guest on set today.
Yes, actually, I brought Bobo with me today. Bobo, would you mind? Oh, yes, Bobo. Hello. Hello, Bobo.
You seem like a very friendly fellow.
Yes. Actually, Bobo is very violent.
Ah.
Yes. He's angry. He's an angry monkey. Sasquatch, actually. But yes, we've created a completely new species. Of course, his name is Bobo. We haven't, as I mentioned earlier, perfected genetic engineering. So, he's mildly to severely retarded. And he doesn't like it when I say that. But it's the truth, Bobo!
You should spell it out next time.
I'm not. Bobo needs to learn. But Bobo is actually very vicious. You see, he doesn't attack me because he sees me as his mother figure. But he will attack any other person who would say wear a mask all the time, you know? Masks offend him a lot.
Would you like to feed Bobo?
Bobo doesn't eat.
Does he not eat? Does he eat a
Bobo.
genetically made pig?
Bobo seems to have gained either a liking or a hatred of you. Oh, hatred.
Oh my word!
Your face is hideous.
Oh my God!
Did you kill him? That took years to make. I guess all we can do now is remove the body. I'm sorry, Bobo. I'm sorry. I had to end like this.
Well, folks, it seems like that's our show, and good night.
Quiz time. Question 1. What is genetic engineering? A. Breeding animals. B. Cloning. C. The process of electrocuting DNA fragments in a gel. D. The study of DNA. If you picked A, B, C, or D, you're wrong. It was secret answer E. The manipulation of DNA. Question 2. How do you transform a cell? A. Inject DNA into it. B. Add restriction enzymes. C. Use a PCR polymerase chain reaction. D. Insert nucleus into egg cell. The answer is A. Inject DNA into it. Question 3. How do you cut DNA? A. Radiation. B. Add restriction enzymes. C. Gel electrophoresis. D. Scissors. The answer is B. Add restriction enzymes.